is in itself an interesting area of research. My second hat is as Deputy Chair for the Steering Committee for the Common Fund Data Ecosystem. So just to point out, the Common Fund is currently funding a number of projects, each roughly the order of magnitude same as MPRINT. And the Common Fund Data Ecosystem is a separate project that is tasked to integrate the metadata, the tools and resources that are coming from all these projects, and to create a cross-cutting discovery platform that makes use of these individual resources, but combined. And in that sense, no surprise, a lot of the conversation I've been having with Sarah and with Lang is, how can we interoperate between the knowledge that's coming out of MPRINT and make it available to the community of the Common Fund researchers? I would like to see MPRINT resources findable, discoverable by Common Fund researchers. And I would also like to see whether it's possible to leverage Common Fund data ecosystem services to augment, to enhance workflows that you have within the MPRINT community. So this is the basic premise. Might be at this stage overambitious, but it is important to be able to explore what are the possibilities. One low-hanging fruit that I'm observing is that, of course, your knowledge base is ripe for interoperability. You have semantic classes that are representing PK parameters, tissue specimens, drugs, of course. And it would be interesting to see whether we can obtain bindings from uniform resource identifiers within the knowledge base and embed it within the knowledge environment for the CFDE. So we have a journey in terms of the type of disciplines that we're going to be meeting over these slides. So I organized these slides over 6 take-home messages. The first being that the CFDE is, of course, a rather young project, about 3-4 years old. And it is, as I said, tasked to cross-bridge Common Fund project data, metadata, and tools. The CFDE right now is supporting primarily discovery, not what you might call development. And indeed, right now, the pharmacometric use case within the CFDE is underdeveloped. And I'm looking to the MPRINT community to perhaps change that. Spark, as a project within the Common Fund, is what I would see as the closest neighbor to MPRINT, given that we are also directly involved in not only the discovery of important neural pathways, but also in the development of medical devices. And in that sense, there's much crosstalk that can happen between Spark and MPRINT. Not just that, but we organize our knowledge about autonomic function in terms of the effect of the autonomic nervous system on peripheral organs. And in that sense, a core part of our map is a flow model across multiple scales that is describing flows across the main thoracics, exchange between main tissue fluids at multiple scales, not only at large membrane level, but also at cellular level and subcellular level. Also, like MPRINT, Spark has a significant modeling component. Of course, from our perspective, we come from a bioengineering perspective, so it tends to be a very kind of mechanistic conservation of mass, conservation of energy type of models. Nevertheless, we rely heavily on modeling for our work. And the last point I'd want to make, and something that I'd like to explore with you, is how can we leverage MPRINT resources to make them findable by the Common Fund community, and vice versa? Can we improve or augment MPRINT workflows with CFDE services? So let's start from scratch. I chose 8 projects within the Common Fund space, apart from Spark, that might be relevant to the MPRINT use case, and I quickly introduced you to them. The first one is Bridge to AI, which is an effort to ensure that clinical data emerging from EHRs is made more amenable for AI ML processing. First one being Chorus, that is coming out of Harvard, that its use case is in decision support for ICU on the bedside for stroke patients. The second one is AI Ready. Its use case is in type 2 diabetes, with particular emphasis on image processing for retinal scans. The third one is rather interesting. It's a voice, essentially picking up key biomarkers that allow us to predict with much more anticipation pathologies that are either neurological, psychological, or indeed respiratory. So one project within the Common Fund is trying to organize clinical data and make it amenable for AI ML processing. Many of you might be aware of Kids First, which is a key repository, a kind of gen-to-fan repository that looks at, from an imprint perspective, birth malformations, pediatric cancer, and rare disease. And of course, Kids First is also leading within the CFDE the use of secure cloud spaces called Cavatica in this case, to help us carry out analysis on sensitive data, but using the latest technology. Some of you in here have already mentioned to me Metabolomics Workbench, which is a key repository that takes lab tests and omics analyses from clinical studies and makes them, annotates them very much along the lines that was mentioned in Dash yesterday. And this is also connected within the CFDE space. MotorPack is interesting. It's an interventional study. It is looking at the effect of exercise on omics signals. It has an animal branch and a human subject branch. And the question is, how do omics readouts change from key tissues in response to exercise? And maybe it may provide a template in the future to understand how pregnancy might impact omics readouts. HubMap, of course, is a crucial resource. It is creating a 3D spatial map of gene expression for both male and female human specimens. One of the key organs that it includes is, of course, the placenta. So it would be useful to leverage these resources to understand what are the omics at play at placental level. And its sister project, SenNet, is looking at how these omics signatures change with cellular aging. So in this case, the combination of HubMap and SenNet can give us some important insight how to use these resources to predict ontogeny models in terms of how receptors, enzymes, and transporters can change, in this case, in the geriatric context, and may have important lessons to learn from a pregnancy perspective. The seventh project is IDG, Illuminating the Druggable Genome. I'm sure that many of you will be familiar with this project. And this project is trying to characterize ion channels, GPCRs, and kinases that are less well understood than most. And in particular, I should like to see a connection between the channels within IDG with the InTech resource that was presented yesterday by Professor Alexounas, to be able to combine models for transporters with channels within IDG in the context of the CFDE. The final project I want to point out is LINX. Now, LINX is interesting. Again, I do not come from a cheminformatics perspective, but LINX is collecting perturbation signatures over a number of standardized cell lines and looks at the effect of a small molecule in terms of the genes that have been upregulated and downregulated. So in that sense, LINX is helping us create a new distance function in terms of the functional effect of a small molecule, so that you can compare small molecules not on the basis of whether one has a Furanose ring and the other one has a pyrrole one, but really in terms of understanding the functional impact of a small molecule on the basis of what genes it upregulates, what genes it downregulates. And LINX has been central in the generation of reprotox style prediction knowledge base that we have generated as a demonstration use case within the CFDE, which I will present in short order. So these were 8 other common fund projects that are being integrated within the context of the CFDE. And the CFDE itself, of course, has its own toolkit. So, for instance, has a visual way to assemble workflows to be able to query our knowledge graph and to be able to find the type of resources that you need. Resources, which I add, would be extracted from the various projects that I have just described. So this is the work of the Common Fund Data Resource Center. And a key part of the CFDE is a knowledge graph, which is using the UMLS as a backbone. And this type of knowledge graph helps us organize and query metadata coming from different Common Fund projects in a biologically meaningful way. So bear in mind, a key objective, a key challenge for the CFDE is to take the metadata models from the different projects and put them together in a meaningful way. So, as I said, right now the CFDE is not particularly active in the context of pharmacometrics, but I would like to present to you 2 projects, each carried out over a period of around a year, and is the result of collaboration from different Common Fund projects, which may have some resonance with regards to the topics that have been discussed during this meeting. The first one is CLOVOC, which is the CFDE's effort to generate a general way to handle clinical data. So in this case, CLOVOC is a collaboration between myself, Deanne Taylor at Kids First, and Shankar Subramaniam at Metabolomics Workbench. And we worked with the TEDDY dataset, TEDDY being the environmental determinants for diabetes in the young. And we used the TEDDY dataset, extracted the information from the codebook, from the dictionary. Incidentally, by the way, resonating with items that were discussed yesterday during the DASH meeting, we only obtained the data dictionary from TEDDY once we got our application approved. So, by the way, if DASH can make the data dictionary available before approval, you are saving researchers so much time. I'm so grateful for this improvement. So we mapped key elements within the data dictionary onto fire profiles after normalizing the data, and then extracted the fire profiles into a knowledge graph. And we focused primarily on key determinants that help us stage patients along the key stages of diabetes, and help us then extract subcohorts from the clinical data that conforms to these key stages. And we used an algorithm that we then developed in-house based upon the presence of first-degree relatives, the presence of a high-risk genotype, the presence of antibodies for particular markers, abnormal GTTs, and of course, clinical symptoms. And we generalized this and created a general tool within the CFDE in anticipation of further clinical data coming, for example, from Bridge2AI, to help us extract, using a combination of arbitrary constraints, any subcohort that we need for our further analysis. So that is CLOVOC. The second project is ReproTox, and this is a collaboration between Lynx, IDG, Spark, and Kids First. So Avi Mayan at Mount Sinai and myself co-conceived this project. Tudor Opria at IDG and Deanne Taylor provided key support and insights into the approach. And a publication is available for your perusal. And ReproTox, in our case, again, this was a one-year demonstration project. So we wanted to show how we can essentially generate new hypotheses for potential mechanisms of teratogenesis. So we wanted to generate a knowledge graph that provides these islands, these cliques, these trifecta of evidence where a birth defect is independently associated with a gene, a small molecule is independently associated with a birth defect, and the small molecule and the gene or the gene product are known to interact. And this approach, again, leveraged the links signatures that helped us find functional neighbors for small molecules that go beyond structural similarity. And the workflow was as follows. First, we started with kits first to extract HPO terms that describe birth defects. And we also extracted potential teratogens from a number of resources, including, of course, Drug Central, using labels that I realize now are outdated for potential teratogens. But hey ho, it was useful. And then we harvested PubMed using natural language processing to find overrepresentation of co-mentions for gene and birth defects and birth effects to small molecules. We also leveraged the combination of TCRD and Pharos to get associations between drug and gene product interactions. We added some further nuance to the likelihood that a gene might be involved in a pathology by carrying out what we refer to as intolerance scores. Essentially, this is based on the fact that in the wild, certain genes have an underrepresentation of mutation compared to the expected amount. And that is suggestive that a gene is more vulnerable to dysfunction if it's altered. And we use that as a kind of biomarker that any interference in an interaction with a small molecule is more likely to lead to dysfunction. We've also extracted data that tells us about which small molecules are likely to cross the placenta to further augment our scoring for putative teratogens. And then we expanded that small list, which was around 200, using the links approach to find other small molecules that have very similar perturbation signatures to make the leap and predict that they might also cross the placenta. And also, we carried out gene-to-gene comparisons, again, on the basis of similar expression signatures from a combination of both of links, geo, and GTEx. And the result is around 500 cliques of essentially mechanistic hypotheses for teratogenesis. And this knowledge graph is available in the public domain. So you can go to the to Avi's site and interact with the Neo4j database and carry out queries, starting with a drug, for example, and looking at what birth defects it might lead to. And the graph will provide a kind of path that links a small molecule to potential gene products that interact with it and the birth defect with which they are associated. And you can also, of course, retrieve the information upon which this information has been based, and you can also extract tables for further analysis and verification. Incidentally, this resource is something that we would like to improve, so I would very much welcome your thoughts and your availability to carry out a standardized feedback process on the ReproTox resource. So Jerry Skeffos, Jerry, can you raise your hand? Is in the room. If you are interested in providing us with feedback on how best to improve the ReproTox resource, we would very much love to hear from you. I also put in my final slide a QR code for anyone who wants to register their interest directly or online. Okay, so now we shift away from CFDE temporarily to focus on Spark. And as I said, Spark, given its direct involvement in development of a therapeutic modality, is the closest neighbor in terms of MPRINT. So this is a schematic of what an electroceutical device looks like. There is right now a huge interest on the vagus nerve, one of the many nerves in the body, but the vagus seems to have quite a deep-seated effect on visceral control. And stimulation of the vagus not only has direct impact on cardiac arrhythmias, but also, for example, in refractory pediatric epilepsy. We're getting some interesting results there. There is, of course, a major anti-inflammatory component that I think links nicely with the previous topic here in terms of microbiome and inflammatory effects. stimulating the vagus does seem to have a significant impact on depression. And it seems that one mechanism here might be through cholinergic-mediated immunomodulation, not only via the vagus direct connection to the colon, but unexpectedly for me, the vagus is innervating the spleen heavily. And the spleen is an important immune center. And there seems to be a mechanism emerging that postprandially, the vagus is stimulating the spleen to reduce the reaction of the immune system to endotoxins that are being absorbed during digestion. And that seems to have an important impact on the muting of inflammation by stimulating the vagus. That seems to have effect even in autoimmune disease like rheumatoid arthritis. This is all conjecture that I'm presenting here, but there is, of course, a weight of evidence that is accumulating. So in order to support the development of electroceuticals, you need a really good map of the autonomic nervous system from the brainstem down all the way to the peripheral organs, to the kidneys, to the gut, to the liver, to the heart, to the lungs. And one reason why we're talking here today is that it would be interesting to see whether this map has any relevance in the modeling of PBPK, PD processes in the body. And of course, I'm not alone in this work. Certainly, a lot of the experimental data being generated by Spark is being processed through the San Diego team led by Marianne Martone. And this is creating a knowledge graph that is then linked to a connectivity model, which I have developed in collaboration with Peter Hunter. And this knowledge is then projected onto a screen to help researchers navigate the map, but also find resources that are relevant to them. And of course, the knowledge that emerges from Spark is being deposited into the Common Fund data ecosystem. So this is a snapshot of the website for Spark. And this is an example of one type of map that we are generating. And by clicking on some particular organ on the right-hand side, up comes the record of the experimental protocol and the data that is relevant. Quite unexpectedly then, HEAL came to us to start reusing and extending this Spark platform now to go beyond the autonomic nervous system, but to start handling pathways for pain, for opioid-driven mechanisms, and also Rejoin, which is focusing on joint pain, is also leveraging the Spark platform to also use our knowledge management system. Also, rather unexpectedly, the acupuncture community came knocking. We had a similar reaction at first, indeed, but I have to tell you, many mothers out there are using acupuncture to relieve back pain, to relieve nausea. And if you look beyond the folklore of acupuncture, there are some significant spinal level mechanisms that are at play here. So if a mother is using acupuncture thinking that this is a sequela-free, complication-free type of treatment, you might want to think again. There are some significant effects that are being recorded, and Spark is helping to understand why. If you buy me a beer, I can take you through the various putative mechanisms, but just bear in mind, it has evolved for some reason, and it is not all folklore. So that is sort of my view from the Spark perspective. Certainly Spark, as I said, in order to articulate and manage all the physiological effects of the autonomic nervous system, we needed to have a model that is attuned to representing physiological processes across multiple scales. And this type of approach has been long in the making. So Peter and I have been collaborating on the development of an open physiology workflow in which we can create a circuit board type of representation, whereby you can plug in minimal models of tissue function into a broader model of advective flow across multiple anatomical sites. And this is early work that is looking at cylindrical structures centered around advective tissues. So this is what we refer to as a functional tissue unit, which is nothing but a biophysical model of diffusion across a number of cells that are centered around a channel of advection that is either carrying out, there's either conducting blood or epithelial fluids. And this work has been of long standing, and it is now at a level where we have essentially a knowledge graph of connectivity that is also easy to query and to visualize. So on the left-hand side, you can see, a classic 3D spatial anatomical representation of the human body. On the right-hand side is a projection of the knowledge graph that we have in Spark. And essentially, it's a homunculus of flow. So in green, you can see the routes of flow for epithelial fluids. So for example, this is the urinary tract, this is the genital tract, liver and pancreas connecting to the gastrointestinal tract here. This is lung, nasopharynx, eustachian tube. In red, of course, is the cardiovascular system, and in blue is flow of CSF. And this knowledge graph is essentially created from many components, many conduits that allow us to then create a layered type of information for anatomical connectivity. So for example, this is a slice, a very idealized schematic slice from the major calyx, looking at the innermost layer for urine, epithelial cell layer, muscular layer, etc. And all these layers are inside, represented in a knowledge graph, mapping to the relevant ontologies. And it also allows us to create modular representations of tissue that you can then connect to be able to track flow of fluids and model flow of fluids using these prefabricated compartments. And here you can see on the left-hand side an epithelial cell straddling the blood compartment and the tissue fluid compartment here in the glomerulus, while on the right you can see an epithelial cell straddling the urinary space and the basement membrane space within the glomerulus. So this is the type of knowledge graph approach to a systematic way to build, to construct compartments within the body, and we want to explore the means of leveraging this type of, high-resolution representation and applying it to PBPK, PD use cases. The penultimate point is really focusing on the modeling side, the modeling of actual dynamic processes. So this is stemming from work that Peter Hunter has been leading in this field, and we recently got an R25 grant with Walter Boron at Case in Cleveland, and both I and myself and Marianne are also involved in this. And this is really looking at a modular way to construct dynamic models in terms of networks of ordinary differential equations for particular units of function within a cell, with particular emphasis on transporters, membrane transporters in the body. And we're leveraging this R25 to work with students as part of their MSc and PhD projects to construct new models with particular emphasis on membrane channels. So this is just a schematic example of a GLUT transporter, but the schematic representation that uses the bond graph formalism to construct these models can then be easily implemented into any language for essentially simulation, given the availability of course, key realistic parameters. But what's key about the bond graph approach is it makes it easy to combine modules for different individual transporters to create more complex representations at a membrane level. And right now, we're well on the way to creating bond graph models for all SLC proteins, and we're of course looking at representing other transporters in the near future. So again, it would be interesting to explore connectivity between what is happening here and the InTech effort that's been described yesterday. So having said that, it is, I think, opportune now to think in terms of what are the avenues to connect MPRINT resources, in particular knowledge resources, to make them findable by the CFD community, and what, if any, of the resources that I have just presented here might be of relevance to the workflows that are happening right now within MPRINT. And I have to say, of course, I'm still familiarizing myself with the MPRINT approach, so I still have much to learn in terms of what is useful and what is relevant to you. So what I would say is that we have a community of researchers who are very keen on exploring new use cases for the resources and the platforms that they are building. Spark is, of course, contributing a model of compartmental connectivity that is annotated to the standard ontologies. And I have been, of course, driving the inclusion of molecular pathway knowledge into the Common Fund knowledge graph. In particular, the Reactome component is now embedded within the CFD knowledge graph. And these components also link to the bond graph models that I have just described, as well as the reprotox resource. So looking at Reactome, of course, the representation of drug ADME is very minimal at this stage. So would one way to improve the relevance of the CFDE to the pharmacometric use case, would it be, for instance, to include something like pharm-GKB that links small molecules to their transporters, to their receptors, and to their enzymes? This is just one suggestion, but essentially what I would like to see happen is that uniform resource identifiers from the MPRINT knowledge base, particularly for drug, for PK parameters, and for specimen, are embedded into the Spark model for compartmental connectivity. And that would then become Spark metadata that is pointing to the MPRINT resource. And the Spark metadata that would be included in the Common Fund knowledge repository, making it findable as part of any search for any individual within this Common Fund space. This is at this stage an idea, but I would welcome further suggestions from the floor and later in terms of what other use cases should be explored. And with that, I think we have covered the 6 take-home messages that I wanted to share with you today. I thank my collaborators, and here's the QR code should you have interest in talking to us about the reprotox resource at further length. And thank you so much. Thank you.
Yeah, that was is the mic on? Yeah, that was a fantastic talk. Really incredible to see some of the things that you've been leveraging from the Common Fund. And I was actually the coordinator for the IDG program and very involved in the early days of HubMap and getting it off the ground. So I can see a lot of potential. But I have two granular questions. So one, I'm going to channel Gil Burkhart from the FDA. Especially as we think about pediatric ontogeny and development. I know you didn't highlight GTEx. There's also a developmental GTEx program. So if you could comment on like how that data could potentially tie in, especially for pediatric ontogeny and modeling. The second, many of these Common Fund data resources like LINKS, IDG, I think the Metabolomics Workbench, they're not active programs now. So the data sets are still there. Have there been discussions about sort of how that data half-life will affect the modeling or, plans to update those kind of resources?
So I'll answer the thank you for the question. So I'll answer the second question first. So the CFDE does provide some minimum lights-on funding to maintain and help map metadata for sunsetting projects. So this is a huge concern of ours. And as you know, the Common Fund has projects that some of them are still onboarding, some of them are sunsetting, others are active. So there are resources to help, especially people with institutional memory of a particular project, help us understand what the metadata means and carry out the appropriate mappings. So there is at least an effort to do that. Metabolomics workbench may have ended, but Shankar is one of the co-PIs on the DRC, on the Data Resource Center for the CFDE. So I don't think that metabolomics workbench is leaving the building anytime soon in that sense. The second question is about GTEx. Absolutely. I'm always bound to do an injustice by merely choosing a sample that I thought might be of immediate relevance to this community. GTEx is an amazing resource. My one impression that I might be sent to be corrected is that GTEx does not do single cell RNA-seq. So it is more of the kind of uber tissue level. So what I wanted to present is HubMap, given that it can help us understand what the molecular machinery on a single cell might be. GTEx may not be yet in a position to do that. On the other hand, the quality of the annotation of a GTEx resource is unparalleled.
Yeah, and so NICHD in partnership with Human Genome has launched the Developmental GTEx Consortium, and there will be at least more inclusion of single cell RNA-seq than in the original GTEx, just because the technology is there. So, yeah.
Indeed, that's very
early days, though.
That's very heartening to know. Absolutely.
Sure. Well, thank you so much. Excellent talk. And 2 questions and a comment. So first is that I saw that I think one of your slides you're showing that you sort of integrate PharmacyKB information to the knowledge portal with all the.
I'm proposing.
Oh, proposing. Yeah, so
it hasn't happened yet. Or...
Okay, great. So I just wanted to actually comment on that if you have maybe interested, if you noticed, there is a pediatric version of PharmacyKB.
I didn't know that.
Okay, yeah, so maybe.
Okay.
Yeah, so that's a specific fund co-funded by NICHD. It's a pediatric version of PharmacyKB.
Yes.
So just wanted to
I mean, the vision there is we already have connectivity with ClinGen within the CFDE through the XRNA group, because PIs from the XRNA, I think Alexander Milosavljevic is also very active in the ClinGen space. We want to bring in these variants that have an impact to PK metrics that are already annotated within PharmGKB and bring them into the CFDE. That is something that I'd like to do. I don't know whether it's needed already, or what are the priorities for the MPRINT community. So I'm here to collect requirements.
Oh, great. And second comment is that you have sort of a great overview of most of the Common Fund initiatives, so that you are sort of are interested in access them to integrate to your research project. So there is another Common Fund initiative called somatic genome editing, somatic cell genome editing, SCGE consortium. I was wondering if you are aware of that.
Not very closely. One of the reasons might be I'm using the diagram provided by CFDE and NIH. It might be that that project has not yet onboarded within the Common Fund data ecosystem. It would be a Common Fund project, but does not yet have a formal connection to the Common Fund data ecosystem. So, and that is something that it would be impossible to mention all Common Fund projects. There are other Common Fund projects, but the ones that I highlighted are the ones that have an agreement to share resources with the Common Fund data ecosystem. So I cannot be eloquent about that particular project. I do not know much about it, but would love to know more.
Well, I can certainly introduce you if you would be interested to the project manager.
Thank you very much. I would appreciate that.
Yeah. Thanks.
You included in your knowledge web, you start to talk about the introduction of flow.
Yes.
And we've found it necessary to look at temporal elements in our knowledge web. So we have a novel approach that looks not only at temporal processes, but also the temporal nature of the information that's in the knowledge graph, because it changes over time. But I was curious because you first talked about knowledge graph based on UMLS, and UMLS very definitively separates out what we call normal physiology from abnormal physiology. And yet that's a process that is an actual time-dependent process, because we have to look at disease as a process, not as states. So I was wondering why you chose UMLS as the basis.
The choice of UMLS did not come down to me. It was basically to find an accessible resource that can be converted into a knowledge graph to help organize queries, given that queries can be coming from so many different directions. It was no attempt to use UMLS to model processes. It was really to create reference points so that people can come in and use concepts that are already present in UMLS to query our database. It is in no way, and it is not, well, I think my glasses want to live on the floor today. It is in no way trying to use UMLS as a single means to navigate and understand our data. So I would say UMLS was not constructed with that use case in mind. Thank you.
I love the timing of this talk, because I think this is an ideal time to interface with MPRINT on some of these issues.
Right.
You know, we've been getting some momentum and getting things done, hitting our milestones and stuff. So now we're at a point where I think this is a conversation that's quite fruitful. But since I'm probably holding up lunch, we'll just make this quick. One, great time for the conversation. Very interested. Two, I think there's some low-hanging fruit here, to be sure, in terms of sharing metadata and things like that. They're kind of no-brainers. Absolutely, there's just some conversations to be had. There's the larger conversation about how to really interact with the ecosystem that you're building here. And I think that's a longer-term conversation. It's going to be more than many meetings and many other stakeholders sitting at that table. But that's a process that I would love to start at lunch if you have time, and we can keep going from there.
The CFDE is moving into, wait for it, phase 2. So it's just launched its data resource center, its knowledge center. Right now, the training center is being evaluated. And there's also an integration and coordination center that has recently been launched. So all change.